Hi, I'm Macy, and today I'm going to be talking about hepatitis A virus, or HAV. HAV is one of the 5 viruses that causes the disease hepatitis, and there are various ways that this disease can be spread. However, HAV is mostly a foodborne illness that is spread through contaminated water and unwashed foods. This virus form is the easiest to transmit, especially in children. So hepatitis A is a viral infection that was first isolated in 1979, and humans are the only natural hosts for this virus. However, non-human primates have been infected in lab settings. So hepatitis A is a non-enveloped virus that has an icosahedral capsid and is approximately 27 nanometers in diameter. It also has a T equals 3 quasi-equivalence, and it has VP1, VP2, VP3, and VP4 structural proteins. So this virus is positive sense single-stranded RNA, and it has approximately 7,500 nucleotides of the positive sense RNA. And this means that it is in class 4 of the Baltimore classification. So for the taxonomic classification, the order is Picornaviridae, the family is Picornaviridae, the subfamily is Hepataviridae, the genus is Hepatovirus, and the species is Hepatovirus A. For the receptors of hepatitis A, they're not typical for picornaviruses. HAV uses HAV cellular receptor 1 to infect the cells, and this receptor is a class 1 integral membrane glycoprotein that has 2 extracellular domains, one being the virus-binding immunoglobulin-like domain, the IgV, and the second being the mucin-like domain that extends from the cell membrane. So the mechanism for HAV to enter the host cell is not known because of the atypical characteristics of HAV, and it cannot be understood from the other coronaviruses how HAV enters the host cell. So after HAV enters the cell, the next step is uncoding, and this is done with the IgV and the mucin domains of the receptors to induce a conformational change in the virus, which leads to the uncoding of the genetic material. So the uncoding stage in the HAV replication cycle is delayed for more than 8 hours, which exceeds the duration of an entire growth cycle for many viruses in the same family. So the cleavage of VP0 as the final step is likely to be necessary for the uncoding of the virus particles in the endosome. So it's possible that HAV can be internalized to a low pH endocytic compartment before the RNA genome is released into the cytosol of the host cell. And the uncoding site of HAV is unknown. So this is just a picture of all the steps starting with the viral entry and then uncoding, replication, and then exiting. So moving on to replication of HAV. The RNA of coronaviruses do not have a 5' cap. Instead, they have a VPG protein that is attached to the 5' end. And the VPG protein is cleaved early in the infection in the cytosol of the host cell. And there is a 600 to 1300 sequence of nucleotides of the non-coding region near the 5' end that is used for the initiation of translation, called the internal ribosome entry site. And the HAV IRES is a type 3, which is different from other coronaviruses. So it requires intact cellular EIF4G to initiate translation. Also, HAV does not shut down the host mRNA translation. It proceeds parallel with the viral translation. So continuing on with replication, picornaviruses use cellular proteins in membranes to establish membranous replication factories that are essentially mini organelles. The membranes are likely derived from the endoplasmic reticulum, and they likely protect viral replication intermediates from being sensed by the RIG-like receptors. And they also provide a platform for the assembly of the multi-protein replication complex. So the expression of HAV2BC and the 2C proteins induces the cytoplasmic membrane arrangements as well as increases the membrane permeability. So 3A, which anchors the ABC to the membranes, has a transmembrane domain that targets its delivery to the mitochondrial domains. And so the replication complexes lie near the mitochondria and the ER. So HAV replication complexes are not dependent upon the phospholipid 4 kinase 3 alpha beta. And the HAV RNA synthesis can be initiated by the guanidine and the cyclosporin A. So next, hepatitis A viruses they likely use the same strategy as other picornaviruses to synthesize new genomes. So a minus strand of the incoming positive strand genome is made first. And the resulting double-stranded RNA intermediate is then used as a template to produce new positive strand genomes. And so the negative strand RNA is in low abundance and it is difficult to detect in infected cells. So the 3D polymerase is the catalytic core of the replication complex, with the minus strand RNA synthesis that is likely primed by uridylated form of the VPG binding to the genomic 3 poly A sequence. And the arrangement of the adenines within the terminal loop means that is likely a slide back mechanism for the uridylation. And the 3' UTR likely adopts a secondary structure that scaffolds the binding and engagement of viral proteins that is necessary for initiating the RNA replication. The positive strand RNA synthesis is primed by the VPG UPU. So next we have assembly. And little is known about the HAV capsid assembly and the packaging of the viral RNA into the capsid. But because HAV has VP1-A and VP4 as structural proteins, it is believed that the assembly process of HAV is different from other picornaviruses. So the 2A in the VP1-A is the primary signal for the assembly of the pentameric structures, which further aggregate to form the empty viral capsids. And when the VP4 is present at the N-terminus of the precursor. So the picornaviral capsid assembly depends on the proteolytic liberation of the individual structural proteins from the precursor P1 or P1-A. And the viral RNA can be packaged into the capsids by the 14S pentameters. So next we have the release, and HAV releases in a non-lytic way that is mediated by the interactions between the capsid proteins with the components of the host cell endosomal sorting complexes required for transport. This is the ESCRT system. So the virions are recruited into endosomes by the ESCRT system to form multivesicular bodies, or MVB, and they are transported to the plasma membrane where fusion leads to the release of the capsids that are wrapped in the single membrane. So for HAV to establish an infection, it has to evade the innate immune system, which is the first line of defense against the virus. So the toll-like receptor 3, the RIG1, and the MDA5 are the main PRRs that are responsible for recognizing hepatitis A. So hepatitis A disrupts the TLR3 signaling pathway by targeting the essential adapter protein, TRIF. And so degradation of TRIF was shown to be caused by the HAV-C or 3CD protein. And HAV can disrupt the RIG1, MDA5, and the TLR3 signaling pathways through the cleavage of essential adapter proteins by two distinct protease precursors derived from the common 3 ABCD polyprotein processing intermediate. So TNF receptor associated factor, or TRAF TLR6 is a key adapter protein in the RIG1 and the MDA5 mediated type 1 interferon signaling pathways. So HAV also disrupts the interferon beta signaling in part through the cleavage of the TRAF6 mRNA via the microRNAs. So HAV can increase the expression of MIR 146A 5P that leads to the cleavage of the TRAF6. So HAV, the 3C Pro cleaves the NF-kappa B essential modulator, or NEMO, and that is the regulatory subunit for the IKB kinase, or the IKK, that controls the NF-kappa B signaling pathway at the Q304 residue, which negates the signaling adapter function of NEMO, and that leads to the evasion of the RIG in the MDA5 and the TLR3 pathways, and thus it inhibits the type 1 interferon response. So the types of infection of HAV. So HAV causes an acute infection that lasts for up to 2 months, and its incubation period is 28 days. However, in some cases, it causes subclinical infections, which is typically in children. The acute infections are more typically in adults. HAV does not cause chronic liver disease or liver damage like the other hepatitis viruses, but in rare cases, it can cause acute liver failure, which is life-threatening. Oops. So next we have transmission. HAV is transmitted through fecal-oral routes, but also the virus can live outside of the body for months, and it is only killed after it's exposed to temperatures that are above 185 degrees Fahrenheit. So it can be transmitted through contact with fomites and then introduction to through the mouth. And HAV has also been known to be transmitted through blood and through organ donation. So here are my citations. More. And that's all.